Hello everyone. In today's tutorial I'll be showing you a few other tools of genomics, and among those, two of the most common tools that we use is UTR selection or untranslated region selection, and then another tool is promoter selection or promoter identification. Okay, so for now, to search for the UTR region or untranslated region, you have to go to the server UTR scan, but unfortunately that server is not working, but I'm showing it to you again because in future it might be available again. So to do so, you have to go to UTR and then you have to write UTR scan, and whenever you are searching for UTR scan, the first thing you see here, the UTR scan CNR is the one you are looking for. So this is the website, and this is the website's home page, and here at the right side you see UTR scan. After clicking there, you are seeing that okay, service temporarily not available. In the normal case, what you will see is here you will have a box here like the other servers, and you have to paste your nucleotide sequence or FASTA sequence here and then press submit. After pressing the submit, it will show you which portion of the sequence is going to be the 5' untranslated region and which portion of the sequence is going to be the 3' untranslated region. So this is the function of UTR scan, and this is the most common website we use for searching untranslated region. So in this part of the tutorial, I'll be showing you how you can search for the prokaryotic promoter and eukaryotic promoter as well. And we'll be using two different servers, not two, basically we'll be using three different servers. One is for searching the prokaryotic promoter and two others will be searching for the eukaryotic promoter region. Okay, so to get the promoter sequence, we need the genetic sequence or gene sequence first, and to get a prokaryotic gene FASTA sequence, we have to go to the server NCBI, and there I have typed RPOE, a popular gene of E. coli. So here you can see FASTA, so I'm clicking FASTA here, and I'll be taking this sequence. I'll be taking this sequence to search for the promoter which is embedded within this sequence. So here I have gone to a web server named Softberry. I'll post the link in the description box. Okay, so for now we can do a lot of works with this software, Softberry. You see, we can find gene for eukaryotes and a lot of other tools. You can see we can even predict the protein structure from this server as well. So for our case, we'll be searching for the prokaryotic promoter, and to do so we'll be using this BPROM, okay, prediction of bacterial promoters. So I'm clicking here. So after going to that server, you have to paste the FASTA sequence. So I have already the FASTA sequence of this particular gene. So I'm copying it, I'm copying the sequence and I'm pasting the sequence here, and then process. And then it will show you the result. So this is the result, and we already know that in promoter we have two specific sites. One is -10 and another one is -35. In -10 we have this kind of Tata box, and these are the sequence of that particular area. So this might be the promoter sequence, and these might be the positions that we are looking for. And also they have predicted 8 promoters for our entire sequence, and they have also predicted some other proteins binding sites as well. Like for LRP, they will bind at this particular sequence where the position of this particular sequence is 3123. Okay, and that's it. This is how we can find the promoter sequence of a prokaryotic gene, and for this case we have got 8. Okay, now we'll be searching for the eukaryotic promoter sequence, and to do so we have to go to the server named promoter2. So promoter2, if I Google it, it will directly take you to the server promoter2. The first result is promoter2 prediction server by DTU, and let's see what we have here. So here we have to paste the FASTA sequence as usual. So here we have the FLS2 sequence, so I'm clicking it here. This is the FASTA sequence for FLS2, though this is the sequence of mRNA, but it will work for us. So I'm copying the entire sequence and I'm pasting it here. You can also upload the file, like I already said to you that we can download the file from this particular area, but for now I am pasting the sequence as the FASTA sequence is not that large. So I'm clicking full output and then press submit. So here we have the entire sequence, and for that entire sequence we have 3 predictions. One is at 300 area where the score is 1.157. So if you have a result which is below 0.5, you can ignore the result. And if you got a score from 0.5 to 0.8, it will show you the marginal prediction. That means it is kind of predicted that promoter might be there. And if you got a score from 0.8 to 1, that means medium likely selection. And the final one is whenever you are getting a score above 1, that means it is highly likely prediction. That means the promoter is there. So this is a server promoter 2 where we can get the prediction of the promoter region, but we can go and search for the promoter by another server. To do so, you have to type eukaryotic promoter database, and then you have this EPD eukaryotic promoter database server, and you can directly search the gene name here, like for our case FLS2. I'm typing it here and then go search for it. And here we know that FLS2 belongs to Arabidopsis thaliana, so we get 2 promoter information here in Arabidopsis thaliana. So if we view here, so this is our desired one, leucine rich receptor like protein kinase. So I'm clicking it here, and this is the sequence. Basically this is where the transcription starts, and in the upstream of this sequence containing the promoter sequence. And here we can get some peaks as well. So these are the region where this particular protein or enzyme can bind. Okay, so this is the area, and we can get the sequence. So this is the entire sequence where we can get our promoter in the upstream of our transcription area. So this is how we can search for the eukaryotic promoter. Okay, so in the future tutorial I'll be showing you some other tools of genomics. Till then, thank you very much.